Hello everyone, welcome back to our digital Western blot video series. Today, we'll explore its practical applications, evaluate its strengths and limitations, and discuss how it's transforming protein analysis through real-world examples. Let's get started with today's video. Digital Western blotting offers several advantages over traditional Western blotting, including speed, accuracy, repeatability, and the ability to quantify results. It is particularly useful in medical diagnostics for its ability to identify disease-specific biomarkers, which are typically proteins that are either overexpressed or underexpressed in certain diseases. Here are some of the most useful and trending practical applications of digital Western blot. Disease diagnosis and monitoring. Digital Western blot is being increasingly used in the diagnosis of various diseases, especially those involving protein misfolding or modification, such as Alzheimer's, Parkinson's, and certain types of cancer. The technique allows for precise quantification of protein levels, helping in both the diagnosis and monitoring for disease progression. Let's take a look at how this technique can be applied in this kind of disease. Alzheimer's disease. The hallmark of Alzheimer's disease is the accumulation of beta amyloid plaques and tau tangles in the brain. These are formed due to the misfolding and aggregation of proteins, which leads to neuronal damage and memory loss. Using digital Western blot, scientists can quantify the levels of these misfolded proteins in cerebrospinal fluid or blood plasma samples from patients. Comparing these levels to healthy individuals can aid in diagnosing Alzheimer's disease. Additionally, monitoring these levels over time can provide insights into disease progression and the effectiveness of treatments. Vaccine development. Digital Western blot has also found applications in vaccine development, particularly can be used for antigen-down serology assays for SARS-CoV-2. Digital Western blot systems offers the ability to use viral antigen ladders to measure serum antibodies reactive against multiple SARS-CoV-2 antigens simultaneously and provide information-rich views into differences in antibody binding patterns across patients. This approach also has promise as an orthogonal confirmatory test to validate results derived from lateral flow, ELISA, or other immunoassay serology techniques. Serological assays load and separate a mixture of viral antigens according to their molecular weight. Digital Western blot is an open platform, and users can utilize any antigens of relevance to their studies for serum antibody capture. Antigens can be recombinant viral proteins or viral lysates. After separation, viral antigens are immobilized using a proprietary immobilization chemistry. After immobilization, serum or plasma is introduced and IgG antibodies in the collected serum or plasma bind to the immobilized antigens. Secondary antibodies like anti-IgG, anti-IgM, or other class or isotype-specific ones are attached to the horseradish peroxidase, or HRP, an enzyme used in biomedical research and testing to be detected by a color reaction or fluorescent tag. Using a mixture of recombinant viral proteins or viral lysates in a serological assay may enable users to distinguish nonspecific interactions and also provide rich insights into the relative binding capacity for several viral proteins in one run, providing increased confidence in results and a deeper understanding of the patient's immune response. The main advantage of using digital Western blot in this context is its quantitative nature. Instead of just confirming the presence of the spike protein, we can measure its exact amount, allowing for more precise control over the vaccine formulation. Furthermore, digital data can be easily stored, shared, and reanalyzed, facilitating collaboration and data transparency. The digital Western blot has other numerous applications in medical diagnostics as well, including monitoring disease progression and treatment response, research and drug development, genetic disorders, and allergy testing, etc. To sum up, its use in disease diagnosis, monitoring, and research holds significant promise for improving patient care and advancing medical science. Currently, One unique product on the market that automates the entire traditional Western blot process is the Simple Western Automated Western Blot System, designed by Protein Simple, now a part of Biotechne. Next, we will refer to the Simple Western Automated Western Blot System as Simple Western for short. Simple Western is the only fully automated Western blot processor on the market. It is a pioneering biomolecular tool uniquely integrates capillary electrophoresis with immunodetection in a fully automated process, eliminating manual Western blotting steps. Distinctively, it covalently attaches several proteins to the capillary wall, negating the need for membrane transfer prevalent in traditional methods. Size-based capillary Western blot assays use capillary electrophoresis sodium dodecyl sulfate or CESDS to separate proteins by molecular weight up to 440 kDa. Analyze small polypeptides or large target proteins like dystrophin. Charge-based capillary Western blot assays use capillary isoelectric focusing or CIEF to separate proteins by isoelectric point. Resolve protein isoforms like phosphoproteins with high resolution. There are several devices in the Simple Western product line from Protein Simple. WES platform's automated capillary electrophoresis for protein separation and immunodetection for up to 25 samples in 3 to 5 hours. Please note that WES has been discontinued as of July 30th, 2021. Protein Simple will continue service and support of the WES platform through July 30th of 2026. JES offers increased throughput up to 48 samples in a single run, faster runtimes as short as 3 hours, and optional charge-based separation and near-infrared or NIR fluorescence detection, in addition to size-based separation and chemiluminescence detection. ABBY performs size-based separation and chemiluminescent detection like WES, but also offers a fluorescent detection option. ABBY is intended for low throughput applications and runs 2 to 8 samples in about 3 hours. Sally Sue and Peggy Sue. These instruments offer increased throughput up to 96 samples in a single run and additional flexibility in assay design. NanoPro 1000. This system offers detailed protein analysis, including post-translational modification characterization with a broad isoelectric point range of 3 to 10 and capacity for up to 96 samples. Remarkably efficient, it requires as few as 25 cells per assay, even for low abundance proteins, saving both valuable samples and time. Simple Western provides a fully automated solution for Western blotting, from sample loading to result analysis. It can be paired with automated liquid handlers for increased efficiency and can process up to 96 samples overnight. The system requires a minimal starting volume as low as 3 microliters and offers high sensitivity multiplexing. Its swift operation significantly reduces the time to results compared to traditional methods. Moreover, Simple Western ensures reproducibility and quantitative accuracy by eliminating the variability inherent in traditional protocols, and the use of total protein assays enhances the accuracy of protein measurements. The cost effectiveness is evident in resource savings, including samples, reagents, wash buffer, time, and labor. With numerous advantages and applications of digital Western blot, it has become an essential tool in modern protein research, helping scientists unravel the complex molecular mechanisms underlying human health and disease. Thank you for watching.